What's up, Aria? How are you doing? First one. What's up, Zenda? How you doing? Your question has a false premise behind it, unfortunately. I cannot accept the conclusion. One of the things about being in shape is there's a variety of things. I am not yoked by any stretch of the imagination. All it requires is basically I would say let me rephrase that. 1 times 4, 5-ish, 5 hours a week for like 2 or 3 years straight, consistently. And that's pretty much it. And eat healthy. That's pretty much it. There's no secret here. There's no secret to being in relatively good health and good shape. If you can commit 5 hours a week, like an hour a day for 4 or 5 days a week, even 3, but you're consistent and you do it week in, week out, month after month after month, that's the answer. That's it. No secrets. What's up, Ahana and Sonia? Nice to see you guys. It's really about consistency. Most things in life are about consistency. Like long-term consistency. That's the unfortunate truth of life. There's no shortcuts. If there were easy shortcuts, everyone would do it. But it's a grind. A lot of things are a grind. Anabolics, they work well for getting like really, really big, but you have to have consistency behind it. Like you can't just take steroids and expect anything to happen. You have to actually do stuff after you take them. What's up, Mr. Chang? How are you? Nice to see you again. Ahana, meiosis is the division of germ cells to create gametes, but mitosis is the division of body cells to create identical body cells, right? That is absolutely correct, as Ashi has reinforced. Mahir, can I go over how to do Punnett squares for blood types with the Rh plus or negative factor? You got it, Mahir. I will do that with let's pull up a Paint. Handy dandy Microsoft Paint. One of the best, most underrated pieces of software that comes standard with any Windows installation, dating back to Windows 95. So this is the problem, right? So a lot of us get confused with these RH. By the way, if you ask a question and I don't answer it right away, I'm either going to get to it or I might have missed it. So you can always retype it as usual. So here, when we talk about like blood types and we talk about like Punnett squares, blood types are interesting because basically when we talk about blood types and we talk about the A, B, and the O, so you've got A, right? You've got B, and you've got O. And you've got RH positive Rh negative alleles, right? These, I want you to, in your mind, think of them as like two separate things. Even though they're both related to your blood type and they determine your blood type, they're two separate traits, so to speak. And so they would be represented by two different genes. So you have a gene, so this is one gene, and this is for another gene, right? And so most of the Punnett squares that we do, we're looking at one gene at a time. We're looking at like, oh, fur color, is it black or white, right? Are you colorblind or not colorblind? This would be the equivalent of like, what is your hair color and your eye color? It's like two different traits. And so what you would do, if you wanted to do a full Punnett square for this, it actually is called a dihybrid cross, and your Punnett square would actually be a 4x4 Punnett square rather than a simple 2x2. Now, if you did them individually, you would do 2x2. So for example, let's say you have an individual who has, let's say you have individual one, let's say we have our female individual, and they have a positive, actually, let's do RH. Let's make it easier because sometimes positive negative is confusing. RH plus and an RH minus. And we want to cross them with a person who is RH, I don't know, let's just say RH. Like the male is RH positive, RH, whatever, just do negative. Two heterozygous individuals. Then we just do a regular Punnett square, like for this particular part. Right, and we know how that goes. You just get the, one allele on top, one allele on bottom. Same thing over here. Boom, boom. And you do your cross, and you find that you get some individuals that end up with two positives, some with positive, negative, and then you've got another positive, negative, and negative, and you got two negatives, right? So that's, and then you would realize, oh, okay, it's 25% chance for offspring to be negative, negative, 25 to be positive, positive, and a 50% chance to be positive, negative. And so when we look at the phenotypes, it's a 25% chance for them to be negative. So this would result in a 25% chance for negative blood type and a 75 per positive, because we know that positive is dominant to negative. Okay. It's written RH plus, RH plus. So, and then you do your A, B, O just the way you've been doing them before, right? So if you did them separately from each other, you could do it this way. But if you wanted to do them at the same time, like if you wanted to take somebody who is regular, like let's just go ahead and let's make a new drawing here. So let's say we have an A. I give you the genotype. There's a person who's A, O, positive, negative, and we cross them with an A. Actually, let's make that slightly different. Let's go A. Sorry, that's not A. I don't want A. We'll go B, B, positive, negative. Now, I would have, if I wanted to do it at the same time, I have to do what's called a dihybrid cross, which means I got to draw this monster 4x4 Punnett square. Boom. Now, you won't have to do this on the test. So this is just an FYI. I won't ask you to do them at the same time. So what you end up with is a 4x4, and then the way you get the sides, the gametes on the sides, is you do the FOIL method, right? FOIL, which stands for first, outside, inside, last. So I take the first, so A and positive, so I get my A positive. Then I do outside, first, outer, A, and then negative, inner, O, positive, and last. O, negative. And I do the same thing for the other one. So you, in this case, you're going to get a B positive, B negative, B positive, and B negative. And then I just do my cross. I go, oh, okay, A, B, positive, positive. That'd be one possible offspring. A, B, positive, negative, another possible. And then we go B, O, positive, positive, another offspring. So on and so forth. And you could just do the rest of the table. I'm not going to spend the rest of the time, but that's how you do it. And then of course you would realize like, oh, okay, it turns out that, you know, you've got like 1/16 of the offspring might be AB positive, positive. Actually, it would actually be 2/16 because down here you also have an AB positive, positive. So this would be 2/16, and you would just do that, and it'd be out of 16 instead of out of 4 in terms of your percentages. This is called a dihybrid cross. I will not ask you to do this on the test. So if I asked you to do a cross, I would probably just ask you to stick with one or the other. Okay, so I don't want to spend too much time on that. Okay. Praval, we're not going to play a Kahoot today. I don't have a Kahoot for this as far as review. Tomorrow, or if I have you, depending on whether you're in my 6th or 7th, tomorrow during class, we're going to do a little practice quiz before the exam, and we'll go over it. So we'll spend about a half hour, 40 minutes just doing a little review with it, and then we'll take the exam. But I don't have a Kahoot today. Today, I'm just going to be answering questions and doing some review. So Mahir, that was the answer to that. I know it's kind of a long explanation, but what else do we got here? Mr. Chang, you're playing Cyberpunk? Nice. I want to play that for sure. I hope you enjoy. Let me know what your review. Give me your review when you've played it enough. Sanjay, during meiosis 1, which of the following is false? A, homologous chromosomes pair up. B, the cells are haploid. You got A, but the answer is B. Can I explain? During meiosis 1. Okay. Let's do this. Let's pull up Google Images here. Find a nice meiosis diagram so we can analyze this. How does this one look? That's all right. That's not bad. Let's use this one. Maybe there's a better one. 1 second. You know what I might do? I'm going to use the slides because I prefer the one that I have on my slides here. I actually like this diagram right here. So your question is basically during meiosis 1. Okay, so here's meiosis 1. Actually, you know what? I like to annotate, so I'm going to exit. Actually, let's exit this. Let's take this, copy it, put it in our handy dandy notepad. Does anyone know what that phrase is from, by the way? Our handy dandy notepad. If you know what that's from, what children's show that's from? Wait, how do I paste? All right, Control C. Oh, it won't let me paste. That's annoying. Well, then we'll just have to use another diagram. All right, this one's decent enough. Okay. So when we look at, so this one, it doesn't have meiosis 1 labeled though, does it? Man, sometimes when you have these, it's really hard to find like a good diagram that's actually like comprehensive, but also not over the top confusing. Very tricky. And I can't find that one that I used It's kind of like this one, but not quite. So, all right. So basically, we'll just use this one. So here's meiosis 1, right? This first part. What happens in meiosis 1 is the homologous chromosomes. In meiosis 1, the homologous chromosomes are already paired up and the homologous chromosomes are being separated. And what ends up happening, the cells eventually get pulled apart, and so you end up with haploid cells. So remember that when you go from a cell that's like this, oops. So let's take a cell. Okay, and in the beginning, you've got a chromosome. You know, here's your maternal chromosome. Let's draw it like this, kind of. And your paternal chromosome. So right now, what you have is a homologous pair because you have one from mom and one from dad. So this is your mother's. So this is maternal. And this is paternal. So whenever you have a maternal and a paternal chromosome in the same cell, we call this diploid. So this, we can label this right off the bat. Let's use like, I don't know, whatever. I'll just stick with black. So this right here is 2N, right? Or diploid. Because it has one from mom and one from dad. Okay, so this is diploid. Now, this is meiosis 1. You end up with 2 cells. Now, what happens is this chromosome goes into this cell and this chromosome goes into this cell. So what's happening is the homologous pairs, which is what this is here. So this is this right. This is a homologous pair. They get separated. And so what we end up with is basically the chromosomes in separate spots. And now, because oops, I gotta switch to blue. Now we no longer have a, right, so I can color these in, whatever. So now you'll notice I no longer have a maternal and a paternal in the same cell. So all of a sudden now we have cells that are N, or haploid. Right, so we can label these as haploid cells. I really need to get a pen to be able to draw some sort of like drawing tablet of some kind, because my handwriting with my mouse is just bad news. So this is N and N, or haploid. So the answer is B, because during meiosis you create haploid cells. Now the one thing, so I see, Sonia, I see you're asking about crossing over. The one thing that I didn't include in this diagram is crossing over, which would happen right here in this phase. So we can label, so one thing we can do is we can just, label this as a homologous pair. Okay, let's bring this down to like, make this a little smaller. So we got a homologous pair there. But what else is happening in this same is the homologous pair has crossing over. So the way I would draw that is before meiosis 1 happens, before they are separated into either cell here, what happens is the crossing over is occurring. So what happens, and I guess I can draw this with like a little, let me just put an arrow here, right? So a little arrow represents crossing over. And crossing over is when they exchange pieces of information. So realistically, if I drew this correctly, what we would end up with due to crossing over is a little bit of the paternal chromosome, the blue, would end up on the red, and a little bit of the red would end up on the blue in some sort of randomized way. It's different every time this happens, but they shuffle up a little bit. And so what I should really draw is showing down here that the crossing over has happened. And so what we have is actually a mix of the maternal and the paternal DNA on these chromosomes. And so this is what it looks like here, right? So this crossing over, right here, when you see this sort of exchange, is happening between homologous pairs, or sometimes we say between non-sister chromatids. And remember, let's label something else here. So these are sister chromatids. So let me sister chromatids. Okay, so you've got two pairs of sister chromatids, but the crossing over happened between non-sister chromatids. So the blue and the red are non-sister chromatids. They are part of the homologous pair. What's up? You asked about independent assortment. Independent assortment is essentially just the only thing you kind of should know about independent assortment. If we look back in this diagram, in this here, right? So independent assortment is this idea that you have basically for whatever gene you care about. Let's say it's gene for hair color, and then there's another gene for another trait like eye color. We can say that those two genes do not get sorted at the same equally. So for example, like maybe it'll help if I draw this. So let's say, don't save this. So let's say you've got chromosome, we'll just make this easy. So here's chromosome number 1, right? Chromosome number 2. Let's use colors here. So this is the maternal chromosome, and we'll use blue for paternal as usual. So one of the things that we can say is, let's say there's a gene, and we'll use like purple here. So let's say there's a gene right here, and there's a gene right here. And this is for hair color. So you've got your hair color gene. And let's say you have a big B for brown and a little b for blue. Or sorry, blonde or something. And then on this one, let's pick another color. Let's go with, I don't know, brown. So over here, you've got another gene. And let's say this is for eye color. And let's say you've got blue, and you also have blue. What independent assortment states is basically that the brown allele, let's say the brown allele for hair color here, right, does not have to go to the same sperm or egg as either of these alleles. Like, it can be any combination. So you could get a brown hair color allele, and I should actually make this more equal. So like, let's make this big B just so it's more clear. It's not a good. So this is big B here. So just because you have a brown hair allele here and a brown eye allele here, these two dominant alleles, they don't have to get inherited together. So it's like not like your offspring always have to have the brown hair and the brown eye together. They could get the brown hair and the blue eye. They could get the blonde hair and the brown eye, or the blonde and the blue. Like it could be any combinations. We say these are assorting independently of each other. It's completely random. That's what it means. So that's kind of what independent assortment means. I know it's a little tricky of an idea, but just the way to think about it is no two genes for a trait necessarily have to be inherited together, or any alleles have to be inherited together. It's completely random. Let's see. What else? Sperm cells sometimes contain the same genetic information. A, sometimes contain the same... Wait. These are true false questions, Sanjay? I'm not quite sure. I actually don't know what the question is saying. Sperm cells sometimes contain the same genetic information. A, sometimes contain the same genetic information. B, are generally all viable. The answer is B. Can you explain? I don't know what that means. What's the question? It's a statement, right? Sperm cells sometimes contain the same genetic information. And then answer choice A is just restating. I don't really get it. I think you mistyped something there. What's up, Nathan? How you doing? Oh, okay, so it's true, false, some sperm cells, and then it's either A or B. So basically what that's getting at is this idea that you have, after meiosis, when meiosis creates the 4 sperm cells, you can never, due to crossing over and independent assortment, you could never get 2 sperm with the same genetic information. Right? So every single sperm is going to be unique to each other. Now, are generally all viable is technically true, because unless non-disjunction happens, any sperm can be used to fertilize an egg and create an offspring. Right? The only exception is during non-disjunction. See, Ashi, for crossing over in meiosis, is it possible for it to occur so that the homologous chromosomes are 50-50 maternal and paternal? That's a good question. I don't know. It's possible, I imagine. I don't know enough about exactly like how the crossing over works to such a level where I would know exactly like what the average percent of crossing over that occurs is. I'm not sure. Amina, can I go over how to figure out which blood types someone can receive? Yeah, I could do that. There's a good website. that does this, but we can draw it out here. So if my blood type is, let's say my blood type is like, I don't know, we can just start with something simple, right? Let's say my blood type is O negative. Okay, so that's my blood type. So the question is like, well, what blood could a person that's O negative receive? And the way to think about it is you have to first determine what antibodies does this person produce, right? And the person will always produce antibodies for antigens they do not have on their blood cell. So a person's blood cell, if this person's O negative, their blood cell does not contain any antigens, because O is the absence of an A or a B, and negative is the absence of the D antigen. Remember, you have the A antigen, the B antigen, and a D antigen. These are your possible antigens and antibodies. O means it doesn't have A or B, and if it's negative, it does not have the D antigen, so there's no antigens on the blood cell, which means it produces the A, B, and D antibodies. So what that means is if it produces all three antibodies, it's going to be unable to receive any blood that has an antigen A, B, or D on it. So it can only receive other O negative blood. So it's considered a universal recipient. Whereas the opposite extreme, imagine a person who's got A, B positive blood. If we looked at their blood cell, you would see that they have the A antigen, the B antigen, and the D antigen. All antigens present. If they have all antigens, then they have no antibodies for this. Oops. So they can receive any blood. So we call them a universal receiver or universal recipient. And this person is a universal donor. What's going to be the ratio of questions to time? It's going to be about like 5 questions. It's going to be, usually you get about 50 to 60 seconds per question. And so that's kind of the ratio, 50-ish seconds per question. So for example, we give you, let's say you have 7 questions. So 7 questions would give you usually about 6 minutes on average. Yeah, 4 parts. 4 parts. I'm bored. Can I go over the difference between sister chromatids and homologous chromosomes? Yeah. So the difference is simply a matter of whether or not they are identical or not. So I actually have a good slide here that I'll show. So the way to think about it is that homologous chromosomes or homologous pairs are basically the same chromosome, but they are not exactly identical. One you got from your mother and one was from the father, right? So it's going to be like a maternal and a paternal version of the same chromosome. So they have the same genes, but different alleles. So for example, in this picture here, you see homologous chromosomes are the purple and green. So the purple could be from, the father, and the green could be from the mother. So they're the same chromosome, let's say chromosome number 1, but it's the mother and the father's version that you received. So they are the same in terms of the number of chromosome, but they're going to have different alleles, right? You might have like an allele for brown eyes on one, but blue eyes on the other. It's what determines your genotype. Now, sister chromatids are identical copies of a single chromosome, right? So after the DNA is replicated, homologous chromosomes are copied, and you end up with 2 of each. These are sister chromatids. So these 2 purple ones are identical, exactly identical to each other. They have the same alleles on each. No difference. And the same with the green ones, are identical to each other. And so what you have here is basically the different colors represent homologous pairs, and the same color represents sister chromatids. Is there an annotation feature for Google Slides? Doesn't look like it. Does that make sense? I'm bored. Kind of a breakdown there. What's up, Sanjay? No, the different stages of mitosis and meiosis are not going to be tested. You need to know that meiosis has 2 phases and like what kind of happens in each phase, but you don't need to know the specifics of what's happening in like anaphase, metaphase, telophase, these kinds of things. Nathan, am I going to play Minecraft, continue Minecraft, or play some Among Us? Good question. I don't know. I'm kind of feeling like either right now. I haven't played Among Us in a while. Maybe we'll play a little Among Us. Kind of mix it up. Ashi, for the test, is it 7 questions per section? No, it's not. Not every section has 7 questions. It's like probably, I think the way we have it right now is it's like 7, 6, 7, 9, something like that. 7, 6, 7, 9, something like that. It's a total of 28 questions, not perfectly divided evenly as 7 apiece. Or 7, 5, 7, 9, maybe something like that. 7, 5, 7, 9, something like that. When does crossing over happen? End of interphase or meiosis 1? End of interphase or meiosis 1. So, crossing over is as soon it's probably closer. It's not. It's definitely before meiosis one. It's definitely before meiosis one, or like before the end of meiosis one. The way technically it's happening in prophase one, so at the very, very beginning of meiosis one, the very beginning of meiosis one. So if we were getting technical, it'll be at the very beginning of meiosis one. Do they show it on here? Yeah, because you see, like in this diagram, right? You see crossing over has happened here before meiosis one. So it's kind of like right at the beginning. So without knowing the individual phases of meiosis, we can just say it's at the beginning of meiosis one. Just the basics, Amina. You don't need to know anything crazy as far as like spindle fibers and how they line up in different phases. Just know that basically like the diagrams that I have on these slides here. So like, you know, like this diagram here on this slide is probably your best bet for what you need to know as far as the steps and phases of meiosis, right? So you need to know that you have DNA replication first. That's happening in interphase. Then meiosis one is when you have this sort of crossing over event between homologous pairs. And at the end of meiosis one, you end up with two cells that have had crossing over occur already and are haploid now, that have half the number of chromosomes because they've been separated. And then finally in meiosis two, at the end of meiosis two, you end up with your 4 haploid cells, and the process of sister chromatids separating is occurring in meiosis two. In interphase, or wait, do sex cells have organelles? Yes, they do. Not a complete set. Egg cells have more organelles, have all the organelles. Sperm cells, as far as I'm aware, they only have mitochondria because they just need a little bit of ATP to power the tail on the cell to push it towards the egg, but it does not, as far as I'm aware, contain anything other than a mitochondria to give it power. Do the number of chromosomes duplicate in interphase? So if you look at what's happening here, the total number of chromosomes do not double in interphase because you go from 2n and you're still 2n after the replication. So you're not actually creating any new chromosomes, you're just replicating the DNA. And that's kind of like one of the more confusing things about meiosis is this part where it's like you start, so let's just take one one chromosome, for example. So let's say you have chromosome number 1 from your mom, so the maternal version of it, and you have the paternal version of chromosome number 1. Boom. Okay. And we can just, just put a cell around it to make it clear. And we can say that goes through DNA replication. And what you get is, what we see when we have them doubled, which is like now kind of like this situation. It looks like you doubled it. Now, to be sure, you now have twice the amount of DNA, right? Because you've got 2 copies of each. But we don't say that you've doubled the number of chromosomes, because these 2 chromosomes here, this one and this one, they're identical to each other, so we don't really count them as another chromosome. They're basically the same thing. So what we go from is 2n to 2n. Even though it looks like we have more chromosomes, we just have double the DNA, but the number of chromosomes stays the same. And this is where a lot of students get confused, because they think, well, shouldn't we go from 2n? Now, shouldn't this be 4n technically, because you have 4 instead of 2? But no, we don't count these 2 as different from each other. So really, it's still 1, 2. And it's 1, 2 over here. Right, so we never count sister chromatids as another chromosome. They get counted the same. So, yeah, that's a little confusing. Is that why it doesn't really go through anaphase when non-disjunction occurs? Because it isn't really a whole cell? Ahana, I'm not actually sure what your question means. Why what doesn't go through anaphase? When you say, is that why it doesn't go through anaphase? I'm not sure what you're referring to. For the meiosis worksheet, is there a key? Which worksheet? Are you talking about this one? These practice questions here? Or are you talking about this one? The review one, okay. This one. I don't know if I have a key, actually. Let me look. I don't think... This was not a worksheet that I made. This was made by another teacher. So I don't have... Let me see if there's a key available. Amina, if we start with 46 chromosomes in meiosis, then by the end of meiosis 1, we have 23 chromosomes in each daughter cell. So in that case, how do we end with 23 at the end of meiosis 1? That's a good question. And again, it goes back to the sort of, we don't count sister chromatids as different from each other. So remember, in meiosis 1, we're separating homologous pairs, right? So it's pretty clear that at the end of meiosis 1, we end up with 23 because we separated pairs. But the only thing that's happening in meiosis 2 is that we're separating sister chromatids. And remember, we don't count those as different. So one copy ends up here and one ends up here, but they're the same. We count them as the same. So you actually don't lose any total number of chromosomes after meiosis 2. Let's see, meiosis crossing over. I don't have a key. I don't think there's a key for this. For this one. But if there's any on here that you want me to go over, I can quickly go over them. You want to go over number 11? also in the mitosis versus meiosis, a term was used, tetrads. Oh, tetrads. Tetrads, yeah, I forgot to talk about tetrads. But tetrad, if I bring up my paint drawing, a tetrad is just when you have a homologous pair that has been replicated. So this here, this is a tetrad. Tetra meaning 4, right? So you have your 2 homologous chromosomes, both of which have been replicated to create 4 sister chromatids in a row. That's a tetrad. No, I'm not going to throw the word tetrad at you. It's just a word to describe the point at which you have 4 sister chromatids lined up next to each other. 2 pairs of sister chromatids, yeah. And usually we only call it a tetrad when they're lined up next to each other like this, and they're doing some crossing over. So crossing over occurs in a tetrad, basically. Right, so it would be a crossing over between like this piece and this piece would cross over with each other potentially. Yeah, I'll go over that for you real quick too. So I'm gonna go over ominous thing real quick, Ashi, and then I'll go over question number 11 on the worksheet. So if we pick up with this same diagram here, the way to think about this is, so I'm going to assume that you understand how this is 2n and this is still 2n, right? Like, so the reason why this is 2n and this is still 2n is pretty much the same reason why we go from n to n in meiosis 1. It's the same reason, but just backwards. Instead of adding one that we don't count, in meiosis 2, we're subtracting one of these 2 that doesn't count. So like if I go forward, let's draw this out then. So let's say we go through meiosis 1. Okay, boom. We'll say this is meiosis 1. Meiosis 1. We get our 2 cells, right? And in meiosis 1, these are separated. So we end up with basically a maternal chromosome here, right? And these are connected. And a paternal one here. Okay, now again, this is now this is n. This is n and this is n. Okay, why is this not 2n? It's because we don't have a red and a blue in the same cell. We don't have a maternal and a paternal version, right? In the same cell. So the homologous pair has been separated. The maternal version went here and the paternal version went here. Okay, now we go boom to meiosis 2. We end up with 4 cells. Get 1, 2, 3, 4. And now what happens in meiosis 2 is the sister chromatids are separated. The identical copies are separated. So we end up with 1 here and 1 here. And then for the paternal one, it's the same. We end up with 1 here and 1 here. Now notice, this is still N. These are all N. N, N, N. Why are they still N? Because remember, these two don't count as two different chromosomes. They're the same. So even when we separate these two and put them into two different cells, we haven't changed the number of chromosomes because these are identical to each other. So there's still only one of this, right? Does that make sense, Amina? Is that more clear now that I drew it through all the way through the process? And yeah, Rakshita, it's going to be 4 multiple choice sections, anywhere between 5 and 9 questions each, for a total of 28 multiple choice questions. Okay, good. Glad that makes sense. It's pretty confusing. It's very confusing, but once you kind of wrap your head around it, it's actually not too bad. It just takes a while for you to kind of make sure you parse it and understand it. So if that's good, let's jump back to question number 11. Okay, question 11. Make a connection. Organisms normally have blank of each type of chromosome, and therefore each gene/allele. One from the mother and one from the father. As a result of meiosis 1, the 2 chromosomes/genes/alleles are separated into separate cells, and only one is passed on to the offspring. Mendel called this the law of segregation. So Mendel is a guy named Gregor Mendel. Gregor Mendel is sort of considered the father of genetics. He was this dude who basically was I think a church, like a priest or something like that. Or he was like a botanist that worked in a church, and he kind of looked at pea plants, and he kind of figured out the whole Punnett square thing. Like he kind of figured out how things were passed from one generation to the next. Yeah, our boy Gregor. He's relatively well known as the father of genetics. So he said that he called this the law of segregation, meaning that things get separated, right? And how is this fact represented by Punnett squares? So first of all, let's go back up to the top here. It says make a connection. Okay, organisms normally have blank of each type of chromosome. Does anyone know the answer to this? Normally have what? How many? It's a number. Yes, 2. So this is 2. Organisms normally have 2 of each type of chromosome, and therefore each gene and allele, right? One from mom and one from dad. Maternal/paternal. So as a result of meiosis 1, the 2 chromosomes/genes/alleles are segregated into separate cells, and only one is passed to the offspring. Mendel called this a law of segregation. How is this fact represented by a Punnett square? So the way it's represented by a Punnett square is, let's make a new one. Okay, so here's organism 1. It's got brown eye and blue eye allele, and we cross that with another organism that has, let's say, 2 blue alleles, right? Now, this is represented in a Punnett square simply by, what do we do when we make a Punnett square? We take, let's say this is the female and this is our male. When we make a Punnett square, what we do is we segregate the 2 alleles, right? We're separating them. We separate them. So what we end up with is now an offspring that is getting only one of these 2 from mom and one of these 2 from dad. So basically what happens when the mother makes the eggs, the egg gets either the big B or the little b. It gets one or the other. So they get separated into different eggs. Same thing with the sperm. The sperm get different egg or different alleles. And so when the egg meets the sperm, and remember, these are represented, this represents a sperm. So I'll draw like a sperm with a little tail, and then these are your eggs. So what a Punnett square is really showing is when one of the sperm that this male can produce, which contains the blue eye allele, were to fertilize an egg that this female could produce, which contains the brown eye allele, you would get a child that gets one brown eye allele and one blue eye, one from mom and one from dad. And then the same thing over here, little b, you get a big B, and little b, right? So each offspring only gets one or the other from mom, and only gets one or the other from dad. Does that make sense? Ashi, right? Wait, Ashi, did you ask this question? Yes, you asked this question. You understand, but you don't understand how to word it? Well, you could just say that when we do a Punnett square, the Punnett square actually separates the alleles, right? It segregates the alleles into different boxes in the Punnett square, right? So notice that if I drew this line up here, right, to kind of represent the Punnett square actually represents the segregation simply by drawing a line between the two mother's alleles and between the two father's alleles. So we separate them in the Punnett square by physically drawing a line between them to show that they're being split apart during meiosis 1. Okay? It's kind of the best as I can explain it. So I'll go over number 6. Yeah, I'll go over number 6, Ahana, and then I'll go over number 12 after that. I'm bored. And then Amina, you asked, for all sex-linked problems that we have done, where it's based off of the X chromosome, could there be a problem where it's based off the Y? No, I would not ask you about a sex-linked that's based off the Y. And for most sex-linked traits that exist in the world, they're mostly related to the X chromosome, because the X chromosome is much larger than the Y chromosome and it contains way more genes. So most things that we care about in terms of sex-linked traits are already on the X chromosome. So that's why we're sticking with it. Okay, so let's do number 6, and then let's do number 12. Let's take a look at number 6 here. Okay, number 6 is just a fill in the blank. So during interphase, before meiosis, the chromosomes are blank. Does anyone know the answer to this? Yes, replicated. That's the best answer to put there. If you put copied, that's fine. Copied, replicated, duplicated. As a result, each chromosome has 2 blank before meiosis 1. Let's see. Has 2. Probably the best thing to put there. There's actually a variety of things you could put there. You could put like has 2 copies or 2 chromatids. Yeah. I would say as a result, each chromosome has 2 copies or 2 chromatids. If you say sister chromatids, it's not true that it has 2 sister chromatids, right? It has, oh no, that's true. Yeah, 2 sister chromatids. That's fine. 2 sister chromatids. That works. I always find it weird that when we talk about cells and like they divide and we talk about the chromosomes, we always use like female gendered language. We talk about like daughter cells and sister chromatids. It's weird that we gender the chromosomes and the cells that are created from this. Wait, wouldn't it be 4 sister chromatids because every homologous pair has 4 sister chromatids? When we say each chromosome, we're not referring to the entire homologous pair, Amina. We're saying each chromosome. So the 2 chromosomes that make up the homologous pair are different. So each chromosome has 2 sister chromatids, but the entire homologous pair has 4 sister chromatids. Brother chromatids. What about bromatids? A couple of bromatids just chilling before meiosis 1. Let's see. What else do we want to hear? What else are we going to go over? Oh yeah, we're going to go over number 12. On board wanted to go over 12. So we'll go over number 12. And then number 9 as well, because Praval wanted to go over number 9. Where's 12? What's 12? What happens to the 2 sister chromatids in meiosis 2? Okay. Well, they get separated. I mean, insofar as like it's just an easy answer there. They get pulled apart. So, actually, I'll just pull up a picture. So if you look at this picture, meiosis 2, you can kind of see that you have each chromosome still has its sister chromatids attached, and then after, those sister chromatids are separated and pulled apart into 2 different cells. Let's see, what else do we got? And then number 9. Oh no, yeah, number 9. Let's see, like number 9. Okay, this is a multiple choice. So Praval, crossing over exchanges alleles between autosomes and sex chromosomes. No, because you can never have exchanges between different numbered chromosomes. Chromosomes always cross over if they're the same type. So chromosome number 1 crosses over with itself. Chromosome 2 crosses over with itself. So a sex chromosome, which is chromosome number 23, would not cross over with one of the autosomes, which are chromosomes 1 through 22. They wouldn't do that. So that's no. Crossing over exchanges alleles between sister chromatids? No. Because sister chromatids are identical to each other, remember. So nothing would happen if they shuffled. It would just nothing. It would be like, imagine having like never mind. That wouldn't, the analogy I was thinking of wasn't going to work actually. So you got sister chromatids, they're the same, so it wouldn't matter to shuffle between them. C, maternal and paternal chromosomes. This is the answer, right? So the maternal and paternal chromosomes, these are chromosomes of the same type, but are not sister chromatids. So they are the homologous pairs crossover. For part D, somatic cells and gametes, no. This doesn't even make any sense, right? So somatic cells are like cells that are part of your body, like your skin, your eyes, everything like that. And gametes are the cells that are a result of meiosis. So part D doesn't make any sense. So, okay. Hopefully that's pretty straightforward. What else? Eva, could I go over number 13? How is meiosis 2 similar or different than mitosis? I'm gonna wait. What are autosomes? Are they like all the 22? Yeah, autosomes are chromosomes 1 through 22. And then the last pair of chromosomes we call the sex chromosomes. It's X or Y. That's the 23rd pair. So Ashi tried to help there. So Ahana corrected herself, right? That's not true. They don't both result in diploid cells, right? Because mitosis does result in diploid cells, but meiosis results in 4 haploid cells. Ashi said they are similar because sister chromatids are getting separated from one another. They are different because meiosis is haploid to haploid, while mitosis is diploid to diploid. Yes, that is true. That's a good answer there from Ashi. Yeah. So the answer here is essentially like pretty much already what everyone sort of said, but yeah. You start with meiosis. In meiosis 2, you go from... Another way that they're similar is that they both double the number of cells. It just so happens that mitosis goes from 1 to 2. Meiosis 2 goes from 2 to 4. So they double the number of cells. They both separate sister chromatids, but they're different in the sense that meiosis 2 results is dealing with haploid cells, whereas mitosis is dealing with diploid cells. And the other thing is that in meiosis 2, the chromosomes that you're dealing with are shuffled. They have experienced crossing over, whereas the chromosomes that you're dealing with in mitosis have not shuffled. They did not go through crossing over. So, Amina, you're talking about some of the problems on the genetic packets, some of the Punnett squares on the homework. Which ones in particular are you referring to? It's kind of weird because, for example, the colorblind ones, they provide a Punnett square to find alleles of the partners of the kids, but the only way to do that would be to find the alleles of the parents of the partner, and that's kind of unnecessary, right? Packet 7, problem 2. Cody and Ann, both with normal vision, have give you the colorblind son. So again, I guess I would just say that in general, don't feel like you need to use every Punnett square that's provided to you. If you feel like you can do this in your head a little bit, that's fine. Otherwise, just use Punnett square to work through problems if you need to write it out. Yeah, I think some of these are unnecessary. Yeah, you don't need this many Punnett squares, for sure. I think it's a little overkill. You do not need to draw Punnett squares. You can draw them on scratch paper if you need to like think something through or like write something out, but you will not be asked to actually draw a Punnett square. For finding a partner's genotype, yeah, it's mainly guess and check. Rakshita, in my opinion, is the test going to be difficult if you are familiar with the material there will be some challenging conceptual questions. If you can do the Punnett square stuff, you're going to be able to do a lot of the basic questions that are going to ask you to like come up with like what % chance would an offspring have this result, right? Like based off these crosses. So if you're familiar with how to do the crosses, like the incomplete, codominant, sex-linked, these kinds of crosses, you'll do really well on that. If you understand the basics of meiosis, you'll do well with that. The challenging part, the difficult part, will be when you deal with some of the more conceptual questions that might ask you to sort of think about the underlying reasons for some of the things that we've been learning. So for example, like, do you understand like what is going on when we say that like a gene or an allele codes for a trait? Like, so how do you, what is the connection between like an allele, a gene, a chromosome, and a protein and a trait? Like, do you feel like you have a good understanding of how all these things are connected, you know, and like what they mean and what that actually results in, right? Or for example, like why, if you have like let's say you have a dominant and a recessive allele, like what does that mean, right? So if I tell you like, hey, you've got big B and little b, right? And this is brown eyes and this is blue eyes, right? And brown is dominant to blue. What does that tell you about the protein that they produce? What does that mean about the proteins involved in this? Because remember, alleles like the B here code for a protein, right? Potentially, right? These make a protein. And so what if this protein, this brown protein, is dominant to this blue protein? What does that tell you, right? Like, do you understand what's going on at the conceptual level? That's where it gets a little more high level and challenging. So that's where I would say it gets difficult. A gene is made up of alleles and is located on a chromosome. You're kind of right. A gene is not made up of alleles, right? So let's say here's a chromosome. Let's use blue. So here's a chromosome. This is a chromosome. A chromosome is made up of genes. So you might have gene 1. You got another gene. Just, you know, I'm trying to just come up with a variety of different colors here. It doesn't really matter. Gene. Right, so you can kind of imagine, you just go down the line here. You've got all these genes, right? These are genes. Okay. So your genes, you have a gene for something like hair color, eye color. These are your traits, right? A trait is the actual physical result of what the gene codes for, right? So the idea would be like, here's a gene, right? Now, let's say I have a maternal chromosome as well. Right? And I would have this gene on the maternal version, right? If I use the same colors, these are all genes here. I'm trying to remember what colors I used, but whatever. You know what I'm saying, right? All these are genes. I think I used something like this here. So now the difference might be that I have a This one here is a big B, and this one is a little b. These are different alleles, right? They're versions of the gene, right? So this might be for brown, the brown eye trait, and this might be for the blue eye trait. So then, going forward, these alleles are instructions to make proteins. And the proteins, I can say instructions maybe. Okay, determine it's think about it as like the ultimate, this is also known as your genotype, right? Up here, so this is your genotype. Actually, let's put this in better. So let's go back real quick. So at the chromosome level, this is your genotype. Let's use, whatever, we'll just use. And then this would be your phenotype. So let's say your trait could be brown eyes. And your genotype is obviously big B, little b. So your genotype at the level of your chromosomes and your DNA tells you what the instructions are, right? And then that creates proteins, which then determine your trait, which are brown eyes. So if you can kind of understand the connection between all of these different terms here, it would kind of help you understand the conceptual thing behind all of this, right? So there's levels of understanding. There are some people, they can just do a Punnett square and they can tell you what the chances are of an offspring with certain trait, but they don't actually understand this whole stuff that's going on here, like the conceptual stuff behind it. Now, sort of riffing back off of what Ashi mentioned, which I think was a good thing that she said. She described something. Oh no, sorry, not Ashi. Arya said, so a recessive allele signifies the absence of a protein or a protein that doesn't work. Yeah, and I don't remember. I think I said that at some point and I know that Arya, you take good notes. I always see you taking a lot of notes. So I had probably written something out on a slide and you had copied it down. But yeah, like that, the idea that brown is dominant to blue in this case. So I have a blue allele and a brown allele, but my eyes are brown. So like, what's going on? Like, what's going on with the proteins that's causing the blue to overshadow, or excuse me, the brown to overshadow the blue? Right? So in this case, what we can say is that most likely, right, the blue allele codes for, a protein that might not work, right? So it's kind of like it's there, but it's not really producing any protein that's useful. And so all that shows up in the trait is the brown protein, the brown eyes. So oftentimes we can think of that brown eyes, that a dominant allele, the reason why it's dominant is because it actually produces a functional protein that's doing something that creates a trait, whereas a recessive allele might not be creating a protein very, like either not creating it correctly or it's just not producing a protein. And so by definition, it's going to be recessive because it doesn't create anything that could make a trait, right? So it will always be overshadowed by an allele that does create a functional protein. So when I say conceptual stuff, all of that is what I mean by conceptual. And I think oftentimes the reason why biology is hard is because people know the surface level, right? People can be like, can tell you the definition of an allele or the definition of a gene or the definition of a chromosome or a protein or a trait, or they could do a Punnett square. But if you ask them to explain to you everything that I just did, like that is like top tier understanding, right? So if you could actually dig down and understand all of this, you're at a conceptual level that really tells me that you not only do you just understand the terms and stuff, but you actually understand how they're all connected and what they mean and all that. So what I just did is what I would consider, top level understanding. And so some questions on the test might ask you some things like around this. And so that's where those tougher questions come into play. So. Okay. Yeah, so I think those were the tougher questions. And then kind of similar with meiosis. So I use this as an example of conceptual questions related to genetics. But with meiosis, also kind of think a little bit about like some of the conceptual stuff, like what's really going on with the chromosomes and like why is crossing over useful and these kinds of things. Let's see. Can I go over 8? I'll look at 8 in a second. Ahana. Yeah, I'll go over 8. Aria, what happens when someone is homozygous recessive? How does the protein function in the scenario like that? Well, it depends, right? So if a person is, let's say, little b, right? In this case, they have 2 recessive alleles. It might be that they just then... So a lot of disorders or diseases are caused by having 2 recessive alleles, right? Because you can imagine if there's like a crucial protein that's doing some sort of function in the body that makes it like function correctly, if in the recessive version of that is a non-functional protein, if you get 2 recessive alleles, now you aren't producing any functional proteins. And so you might have a disorder, right? A disease or something that's going wrong with your body. And so I remember that someone had asked about like why incest is considered not great. And that's one of the reasons, is because if you have 2 people that are related, they're both heterozygous, it's likely that because they both have recessive alleles, the chance of them having a child that contains 2 recessive alleles that could be homozygous recessive is much higher than if you have a child with someone who is not genetically related to you, because it's less likely that they'll have a recessive allele for a particular trait. And so typically the reason why we say like it's not so great just biologically, right, to reproduce with someone that you're really closely related to genetically, is that you can generate offspring that are more likely to have genetic disorders due to this idea of having 2 recessive alleles that are producing a malfunctional protein or no protein at all. Now, in other cases, it doesn't cause a disorder, right? Like for example, with eye color, if you have 2 blue alleles, let's say, you might just produce, the protein might just be like a lighter version, so your eyes just look lighter than you would with brown. Like with colors, it's a little different, right? Because you can imagine, right? Let's say one allele, this little b allele produces a color that looks like this, right? And the brown allele looks like this, right? Or yeah, like a darker brown. All right. If they were to mix together, you can kind of imagine that just the way colors work, a darker color will tend to like overshadow a lighter color, right? And change the eye color. So for eye color, it's probably more like similar to something like that. Can we go over 8? That's what we needed to do. Describe the process of crossing over. You know, I mean, you don't need to know too much detail. Like this process of crossing over, you might want to be able. You should be able to use the terms homologous pairs and chromosomes. So I would say probably something like a good answer for something like this would be like the process of crossing over occurs at the beginning of meiosis 1, in which you have two homologous we have a homologous pair that exchange genetic information between each other, and the maternal and paternal versions of the chromosome are shuffled up to create new combinations, and this is occurring between non-sister chromatids, something like that. Where's Among Us? That's a great definition, Ahana. That's a good one. Okay. If people want to play, we can jump on and play a little bit before we call it a night, now that most people have got their questions. And if you still have a question that pops up as we're just hanging out, you can throw it in the chat and I'll take time to answer it. See if we can see how many people are out there willing to play. If we don't have too many, then maybe I'll just kind of hang with Minecraft. Right, looks like we only got 3 people. 4, 5. So while we wait for people to join, I am going to use the restroom, and I will be back in 1 second. I also think I want to change my hat. I don't know if I'm a fan of the banana right now. What do I want? What do I want? Should we get in the Christmas spirit? Get the Christmas stuff going on. Christmas tree, Christmas lights. Snowman? Yeah, we'll go Santa hat. Keep it real. All right, I will be right back. We got 7, nice. Hopefully we can get a couple more. All right, we got 8. That might be good enough to play a game here. 2 imposters for 8 people. Okay. Let's do it. Oh, hey, we got 9. We got 9, we got 9. Do I hear 10? Do I hear 10? Do I hear 10? Oh, Sonia keeps disconnecting you. Bummer. Well, we're going to get started. You can maybe join in next round. Remember, no cheating, no sniping of the stream. Where are we going? We're going oxygen room. Get rid of this trash. Go to nav. Okay, we got blue, 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 blue. Coming that way. Download some files. We gotta fix the lights. Complete. Oh, I saw that vent and then it got busted. That's quick. Alrighty. Green. Little double kill action. Wait, who was it? It was Kamisato. They got no info. Wait. Diaz, your game is glitching? Cool Bear left the game. Okay, that's a bummer. Let me... All right, let's do some tasks. What's going on here? I didn't see that kill. That must have been really quick. Mario Judah. That's what happens when you go and get water. No, you literally saw me walk up to upper engine. Wait, did you vent in security? I've got this one on both my eyebrows. I have one or two like couple hairs that just grow super long that I have to trim. I think that's what happens when you get old. Like random hairs just start growing, like for no reason. They just start getting longer, just out of nowhere. And then sometimes I just pull it. This isn't looking good. I don't like the odds of this because we've already lost 3 people and they're none the wiser at this point. And task completion is still really low. Not good. Bodes not so good. The imposters are gonna take it here, I think, unless someone makes a goof Unless one of these imposters really goofs and reveals their identity accidentally with like a kind of a obvious kill. Oh, honey, you want me to play a little Minecraft, huh? Yeah, we'll see. Let's see what we got. It's yellow from Diaz. How are they voting? Are they gonna? Did Diaz and Kamisato just totally convince? So, but they vote. If they vote yellow, it's over. No, wait, is it? Yeah, it's over. If they vote yellow, it's over. Wait, why is Kamisato grayed out here? Anuva, were you Kamisato? Game, it's over anyway. Actually, no, game isn't over because you got kicked. So you got kicked, so now they have a chance, actually. If you hadn't got kicked, it would have been game over. You getting booted was a savior. I don't even know why I'm doing tasks. There's just no way. Yeah, I knew it was a game. Well played, well played, well played. Good stuff, good stuff. Anyone else want to get in before we start for round 2? We've got 7 this time. I think 2 imposters for 7 people is just a little rough. But also like 1 to 7 is rough too. But no, we don't have enough people trying to get in on this, so I guess we'll just go with 7. See how it goes. This might be too easy for the imposters. Might be a little too easy. Oh, there we go. We got 8th. I think 2 to 8 is kind of bare minimum. I think a 1 to 4 ratio, in my experience, seems to be like kind of the minimum. Anything below that, like 1 to 3, it becomes a little too easy for the imposters. Just chill, but I actually got to be seen. What's up, Matt? A little math homework. Makes sense. Did someone see me bent? Oh no, Blue. Why did you do that, Blue? Blue, you can't vote. You said you were going to skip and then you voted for the prime sus. We got to get... So we got to frame... Can we frame Lime here in some way or another? Look at Lime. Completely on me and I don't want to... Look at these guys. I'm going cams. How would she have seen me from cams? I'm trying to use purple here as a little bit of a shield. Comms are sabotaged. That was messed up. I should have gotten... That was a fail. Actually, I think I needed to have gotten a kill on that one before they called this. So what do we at? We have... There's two... If we do a... If we can coordinate a double kill right now... What's up, Drew? We got to do a double kill here. A double kill in this game. That's it. Double kill, we win. Yeah, independent assortment is essentially the idea that you have that genes, right, are not inherited together, right? So they're not like linked. So for example, or rather alleles are not linked. So for example, like I don't have to inherit like the brown allele for hair color from my mother, and the brown eye color from my mother. Like I could inherit brown hair and blue eyes, or blonde hair and brown eyes. Like those two things, those two traits are completely independent of each other. I can inherit them in any assorted way. They don't always have to go together. That's what independent assortment is basically trying to get at. Does that make sense? I know that's kind of a weird idea there, but yeah. That make sense? Got him. That was I don't know how that I got lucky with Pink, Matt. Are you saying that when Pink ran out of the electrical that she went, or that it went right? They went towards navigation, or excuse me, they went towards the reactor rather than towards storage, so they didn't see my kill. Yeah, that was pretty lucky. But I mean, even if they did go that way and see the kill, I mean, there's no way that she or they, I don't know, there's no way that they knew I was coming from storage or coming from the reactor side. Like, it would be impossible for them to tell. I'd be sus, but I wouldn't be guaranteed. Okay. Let's play one more. It's 9. So I think we'll play one more game here. Anyone who's watching, feel free like joining. I don't know how much longer I'm going to stream. It's getting a little late. I didn't know there was a new map. There's a new map that comes out tomorrow. I'll be honest, I actually haven't played one of the current maps. I've only actually played this map, like the original map, and the second map. But I have not played that third map that they have. So I actually should probably even play that one. All right, we're gonna go. We're gonna do a game here with 8. Night, Ahana. Have a good one. Called Airship. Might have been Sketch. Where is everybody at? Oh, shoot. How many sets of chromosomes does a germ cell have? How many sets? They have... How many sets of chromosomes? They have 2. So like a germ cell is like any other regular cell. It's a diploid cell, so it has 2 sets of each chromosome. So it'll have like a maternal and a paternal version of chromosome 1, and then a maternal and a paternal version of chromosome 2, and so on and so forth. Dang. see what we can do here. Oof, risky play here. Yay. Wait, who else was standing there? It was, okay, so it was basically, so basically it's got to convince, they have to convince Amina, green. Or no, wait, green, no, they have to convince brown. So if, it doesn't actually matter. I think it's going to be a tough one to win. So right now, basically, who's going to do a better job convincing the other two, black or white? Brown hasn't really said anything. He's been very silent in this entire thing. I vote white. Also black. Who is the other imposter that vented? Switches. I'm guessing they're gonna go white. Yeah, it's game. GG. I thought we might. It looked like it might have started off decently, but we had that double kill. If Diaz hadn't spoiled it and walked in on us at that moment. All right. I think I'm good. I think I'm good with this. Among Us is, I'll tell you this much. I'm losing my interest in Among Us over time. Now how do I exit out of here? Just this? Leave game. Alrighty. What time is it? It's 9:09. So I'm going to call it a night, I think. But I hope everyone got their questions answered for tomorrow. Keep in mind that we are going to do a little bit of review prior to the test. So if you have questions, you have a chance there to get a question answered. But other than that, have a great rest of your night, everybody. I will see you on, when I see you, tomorrow or Friday. And we'll get it done. All right, peeps. Have a good night. Appreciate y'all. Take it easy.